My name is Matt Hill.
And I'm Alex Kopicki, and you are listening to The Wild Scientists, a podcast where we discuss current events happening in the scientific community today. Wild Scientists. Today, we will be discussing the article Using Hominin Introgression to Trace Modern Human Dispersals written by João C. Teixeira and Alan Cooper. The article discusses anatomically modern humans' migration patterns, who from now on we will be referring to as Homo sapiens. And then, we will be discussing interbreeding that took place between Neanderthals, Denisovans, and Homo sapiens. We will also elaborate a little bit more on this in the podcast later.
To give some perspective, before we begin, the time period we'll be discussing is around 55 to 50,000 years before present. In this case, before present means before 1950.
The signatures found in genetic markers display how introgression remain preserved in the genomes of modern-day populations of humans.
Genetic markers are essentially signals that are found in the human DNA that show ancestry in this case. So it will show like what percentage of Denisovan, Neanderthals, or whatever each person has in them.
This also provides evidence of the sequence and timing of these early migrations, or where and when these migrations occurred. It also proves that Asia was a seemingly more complex area than recently perceived. The article mainly focuses on the Homo sapiens introgression with three main hominin groups. These groups again include Neanderthals, Denisovans, and the newly discovered mystery hominin, EH1, and EH2, which we will be discussing in more depth later on in the podcast. Multiple of these interactions and interbreeding events happened along what is known as the Wallace Line.
The archaeological record and fossil evidence show that these hominids did not travel as far as New Guinea, Australia, and Tasmania. Please note that just because certain populations have traces of EH1 does not mean that interbreeding occurred. In these places, in the place that those modern humans now live, Wallace's Line still is a place where recent genetic evidence found still remains a mystery.
With all of this information, what do you think, Matt, are the three main takeaways from the article?
I would have to say migration patterns, EH1, Homo sapien DNA, and the Wallace Line are essential focal points to fully grasp what is being said in this article.
Yeah, agreed. Starting with these early migration patterns, who do you think traveled where?
Well, to simplify the statistics, Neanderthals hit Europe, hit parts of Asia, and mingled there, in reference to interbreeding. According to the article, the first of these events occurred between a small Homo sapien population and Neanderthal populations somewhere in Western Eurasia around 55 to 50,000 years before present, and created a genomic signal of about 2% introgressed Neanderthal DNA that is now found in non-African populations. Introgression refers to the transfer of DNA that has been combined, hybridizing the individual's gene pool. Denisovans stayed within and around the Asian continent and migrated south into India and Australia. After that, they seemed to have stopped due to other hominin populations already inhabiting that area. One of the Denisovan groups seemed to have settled in that area. Pertaining to EH1, it seems that they mingled with the Denisovan group, which is why we can find EH1 genetic markers in the DNA today.
EH1 was found when researching Denisovans, correct? If I recall correctly, they were genetically roughly equidistant to Denisovans and Neanderthals. Equidistant means that they were so far apart from both Denisovans and Neanderthals that they were basically their own taxonomy. It was detected in groups such as South Asians, the Andaman Islanders, and Aboriginal Australians. The authors hesitantly decided to position the event in northern India.
Yep, the methods used in the study to find these gene markers authors are known as approximate Bayesian computation and deep learning method. You know, ABCDL, with extensive demographic modeling. Basically, the main takeaway is that the authors concluded using these methods that the distantly related Denisovan signal is actually so genetically divergent that it is equidistant to both Neanderthals and Denisovans and probably represents an entirely new hominin group. This is in reference to EH1.
What is interesting about this that the article mentions is that the ABCDL study was able to identify that the AMH ancestor of all Asian and Australopapuan populations interbred with EH1, resulting in a shared genomic signal of 2.6 to 3.4% EH1 ancestry. It also describes that following this initial event, it appears that a second phase of mixing occurred between the ancestors of Australopapuans after they split from other Asian populations and a Denisovan group more closely related to the sequenced Altai individual, generating an additional 1.6% genomic contribution. This basically means that there's a mixture of genes that show evidence of EH1 interbreeding with Denisovans. It proves Denisovans were not the only pre-human groups in the area.
This is a pretty big discovery, since without prior knowledge of EH1, we would have just assumed that Denisovans were the ones mainly dominating this area.
Continuing on our third and final point, Wallace's Line. What's the deal with that?
The Wallace Line is a figurative line drawn in 1859. This line separates a transition zone between Asia and Australia. This is important because west of the line, one can find many Asiatic species, and to the east of the line, you can find a mix of both Asiatic and Australian species. This is important because species do not tend to cross over this figurative line, meaning that species tend to stay on the west or east side, respectively.
Huh. So why is this line so important to the article, do you think?
Well, one of the interesting aspects about the Wallace Line is that there's a noticeably higher amount of Denisovan DNA content observed in the modern-day populations across the Wallace Line. What's so interesting about this is that there were other hunter-gatherer groups located in this region before the Denisovans arrived. This means that not only did many Denisovans decide to make this area their home, but they also expanded and spawned among the already existing populations, which in the end would cause the Denisovans to leave a much higher DNA marker than the other groups such as Australopapuan. One very unique feature about this is that in the Philippines specifically, it is thought that there was heavy interbreeding taking place between the Denisovans, leading to an abnormally high Denisovan DNA marker than most other places.
There are also signs that other Denisovan integrations popped up east of the Wallace Line. However, the data for this is not as strong. The article ultimately ends with the message that a lot more research is needed to gain a more coherent understanding. What we do know for sure is Homo sapiens first moved through Asia and interbred with EH1 in South Asia. The Denisovans in the Icelandic, Southeast Asia, the Philippines, and possibly with EH2 in Flores, which we will be discussing next week. Noticeably, however, the Denisovan of these hominin groups are the only ones known.
It is crazy just to think how far our technology has come that we have been able to find all of this valuable information. Considering most think 23andMe is as far as we'll go to find out more about ourselves and our past, there's so much more to our story.
So true. Hopefully, in the future, we can one day see traces of Denisovan, EH1, and Neanderthal genetics when looking at the heredity websites. Wouldn't that be something?
On that note, this has been a brief discussion on using hominin introgression to trace modern human dispersals. Thank you for coming and listening to The Wild Scientists. I'm Matt Hill.
And I'm Alex Kopicki, signing off.